Hey everyone, today's set of notes will be focusing on a concept known as karyotypes. The things that you need to be able to do by the end of this set of notes is be able to understand what a karyotype is and how scientists are using these to visually represent chromosomes and use these to then identify any abnormalities in chromosome formations in humans or other organisms. We'll also touch on the different variability in chromosomes across different organisms in terms of the number of chromosomes that they have and also the size. And we'll do a couple of activities in class to consolidate what we've spoken about today as well. So to help us with determining the genetic makeup or the structure of an individual's chromosomes, scientists use these images called karyotypes. So karyotypes are digital images that scientists construct when they take a snapshot of the chromosomes within a cell during metaphase. Okay, so scientists will look at cells undergoing mitosis and freeze those cells when they hit metaphase so that they can see the chromosomes in their sister chromatid form. They'll take a digital image of that and then use some digital imaging software on a computer to manipulate it. So they're taking an image that looks like this through the microscope. They manipulate it on their computer and they're able to line up chromosomes in homologous pairs from largest to smallest so that they can see the basic structure, or I guess the basic outline of what the chromosomes in that individual looks like. So as I was saying, those chromosomes in a karyotype are ordered from largest to smallest in their homologous pairs. And we always have our sex chromosomes right at the end. So you can see in the two karyotypes that are shown on this screen, so this one here and this one here, those sex chromosomes are left to the end, regardless of what size they are. All the other chromosomes, or the autosomes that are in these karyotypes, are ordered from largest up the top here and here to smallest down at number 22 down here. Looking at these karyotypes here, just while we're looking at this screen, there's a couple of things that I can determine looking at these karyotypes. First of all, I can tell that I'm looking at humans, so the species of the organism that I'm looking at, because I know that humans contain 46 chromosomes. So if I was to look at a karyotype of an organism that might have had, say, for example, 78 chromosomes, that would indicate to me that, one, I'm not looking at a human karyotype, and two, if I know the exact number that a particular species has in their karyotype, for example, I know that 78 chromosomes appear in dogs, I could make the assumption that I'm looking at a dog karyotype, not a human karyotype. Another piece of information I can get from these karyotypes is the sex of the individuals. Again, I know that I'm looking at humans, and I know that in humans, if an individual has 2 X chromosomes and no Y chromosomes, as we can see in this individual here, they must be a female. And the one on the right here has an X and a Y chromosome, so they must be male. Okay, so, whoops. Female, male. All right, let's have a look at this example here. Do you think it is a male or a female? Hopefully you have selected female, and we can determine that because they have 2 X chromosomes and no Y chromosomes are present. Remember, it is the presence of the Y chromosome that determines whether an individual is biologically female or male. Obviously, if the Y chromosome is there, then it's going to be biologically male. Let's have a look at this one. So is this one biologically male or female? Again, looking, we can see that they have an X and a Y, so they would be biologically male. Okay, this one here's a little bit different. So I'd like you to, first of all, determine the sex of the individual and then determine what is abnormal with this particular karyotype. So looking down at the sex chromosomes here, I can see that there is no Y chromosome present. So that automatically tells me that this is a female, a biological female. But while I'm looking at this, I'm noticing that there's actually only 1 X chromosome. There's no second sex chromosome. This particular disorder is known as Turner syndrome, and we'll learn a little bit more about that in class. Okay, again, let's have a look at this karyotype. what are we noticing that might be a little bit abnormal here? Also, can you determine the sex of this individual? So yes, we can determine the sex of this individual. Looking at the sex chromosomes in the bottom right-hand corner, I can see that they have no Y chromosomes and they have 2 X chromosomes, so they would be female. I'm also noticing that there is an abnormality here with the homologous pair of 21. Okay, it's not a homologous pair, it's actually got 3 copies of chromosome 21. So this is known as trisomy. So tri meaning 3 copies of chromosome 21, or most of you will know it more commonly as Down syndrome. So this is what a karyotype of a female who has Down syndrome would look like. Okay, this one here. So again, what is the sex of this individual and what is the abnormality that you can detect? So we can determine the sex by looking at the sex chromosomes, and I can see that there is the presence of the Y chromosome. So it is a biological male individual, but the abnormality that I'm noticing is that there are 2 X chromosomes. So this individual actually has 3 sex chromosomes, not 2. This particular disorder where they have 2 X chromosomes and a Y is known as Klinefelter syndrome. And again, we'll learn a little bit more about this in class. The karyotypes that we just looked at that were showing disorders or genetic disorders, the ones that we were looking at were Turner's, Downs and Klinefelter syndrome. So the 3 of those were specified in your study design, and we will discuss these further in class, but I quickly wanted to recap why these happen. So these are caused or examples of aneuploidies, and aneuploidies are examples of genetic disorders where one chromosome is either missing or an extra one has been added to the total. So in humans, we might be looking at an example of when someone is missing a chromosome. So instead of having 46 chromosomes, they have 45 chromosomes. Or they might, instead of having 46, they've got an extra one in their karyotype, which leads to having 47 chromosomes. The final set of karyotypes that we're going to be looking at are karyotypes that again are using or are displaying a different sort of genetic disorder. So we've just looked at aneuploidies. These particular karyotypes are showing, I guess, a genetic difference which we refer to as a polyploidy. So a polyploidy is when we're looking at a karyotype or the genetic makeup of an organism and we see 3 or more full sets of their chromosomes. So if we take a look at the image on the left-hand side of the screen here, this is showing you some examples of polyploidy actually in a mosquito. So if we look at the first little part of the image here where it says haploid, this is what the mosquito's sperm and egg cells would look like. It has half their genetic makeup in it, so a copy of every chromosome. Mosquitoes only have 6 chromosomes in them, so half of 6 is 3. The second part of that diagram, the diploid number, we know that diploid means 2. So in a mosquito's regular cell, so not its sperm and egg cells, we would expect to find 2 copies of every chromosome. It would be diploid, so 2 copies, meaning 6 chromosomes in total, and that's what we would expect a regular mosquito to contain. The other 3 images that you're seeing on the left-hand side of the screen are showing you genetic abnormalities or genetic disorders that are displaying a polyploidy. So you can see in this first one here that's labeled triploid, this is actually a mosquito cell that instead of having 2 copies of every chromosome, it now has 3. And that's because an error has occurred when the sperm and egg cells were forming and have caused this organism to now have 3 copies of every chromosome rather than 2. You've then got the more extremes here, the tetraploidy, which is where you have 4 copies of every chromosome inside each cell, or hexaploidy, where you actually can have up to 6 copies of each chromosome in every cell. So lots of different organisms can experience this, not just things like mosquitoes. Polyploidies are super common in, I guess, plants. So things like bananas and strawberries, they have polyploidy, and that's usually what gives them their extra flavor that humans have selected for. You can also find polyploidies in things like frogs and reptiles as well. And I have included a karyotype here on the right-hand side of the screen of a polyploidy that has actually occurred in a human. So we can see that in this human karyotype here, this human has 3 copies of every single chromosome. So not just a third copy of one chromosome, but 3 copies of every single chromosome. Unfortunately, when this does occur in humans, usually the embryo will die in utero, so before it's even born, or if that child does happen to be birthed, these humans do tend to die within the first year of their life. Obviously, polyploidy isn't necessarily a disadvantage to all organisms, just like the examples I provided you before, things like bananas and strawberries, our fruits, our plants, our veggies. Lots of those actually benefit having a polyploidy because it enhances their flavour, it enhances the nutrients that they're providing, and us as humans are seeing that as a desirable trait, and therefore we keep breeding these particular plants to have that polyploidy to, I guess, exploit that feature of them.